I believe it will be very exciting because now we are going to talk about science itself, and it will be 3 scientific projects, presentations of CLARA research programs. And we have got 3 representatives for research program number 1. It will be engineer Petr Kouba from CIIRC CTU with the topic Learning to Engineer Protein Flexibility. The second speaker for research program number 2, it will be Ninon Burgos from Paris Brain Institute. And for the third one, Jean-Marie Bouteiller, who is known for all of you already. So if I can ask, please, Petr Kouba to come here and give his presentation.
Thank you. Okay, hello everyone. So my name is Petr Kouba, and today I will be talking about our work on studying protein flexibility using machine learning and the relevance of such approach for the study of neurodegeneration. So firstly, let me give you a brief introduction about proteins. So proteins are are biomolecules with complex structure, which we can see an example of denoted in green in the lower corner. But interesting property of the proteins is that all the natural proteins are composed of just 20 natural amino acids, which we can see in the table at top. And because of this property, we can also actually represent the proteins in a much simpler way than using their structure. And that is by using the protein sequence. So we can actually represent the protein just as a 1D sequence of the amino acids that the protein is composed of. But why to study protein? What makes them important for human health? So I think many people actually, when they hear about proteins, think of like nutritional supplements and so on, and they're not wrong. I mean, proteins are really important nutrients in the human body. But what makes proteins even more interesting are all the different functions it can perform. And for example, there's important class of proteins called enzymes, which catalyzes chemical reactions. So these enzymes, they can take on the input a molecule, perform a chemical reaction on top of it, and they can output a completely different molecule. And this can play a role in many important processes in nature. Another important class of proteins is, for example, antibodies. And we have an illustration here of the antibodies attacking a virus particle. And antibodies, they're actually the weapons of the immune system, which are used to fight antigens such as viruses. And another role of proteins is that often they can be considered the drug targets. So imagine that in the human body there can be a protein which malfunctions, and this inspires us actually to design drugs which would correct the behavior of the protein, and by this means we could treat the condition that the malfunctioning protein causes. An example of a drug target relevant in neurodegeneration, in particular for the Alzheimer's disease, is the apolipoprotein E, short ApoE. The function of this protein is that it transports lipids to the cells in the body. And interesting thing about this protein is that it can exhibit certain mutations. So most of the population has a protein called ApoE3, but some people in the population can have a mutation at this protein, which makes them the carriers of the ApoE2 protein. And there's also a different mutation, which and the ApoE with the different mutation is then called ApoE4. And it has been actually observed in the clinic that depending on which of these variants of the ApoE protein a person has a strong effect on the risk of that person later suffering from Alzheimer's. So actually, most of the population has ApoE3, but the people who have ApoE2 mutant, they are less likely to suffer from Alzheimer's. And on the other hand, the people with the ApoE4 mutant, they're actually much more likely to suffer from Alzheimer's later. So this observation actually motivates us to study this protein, because if we can understand the differences between these variants of the protein, it could help us understand the disease and design drugs. Our colleagues from ICRC in Brno are studying this protein a lot, and they already released 2 manuscripts studying the process of ApoE aggregation. That is a process believed to be relevant for neurodegeneration. In particular, they studied the effect of potential drugs for the Alzheimer's disease on the process of ApoE aggregation. So I hope now everybody is motivated to study proteins and ApoE in particular. But let me tell you a bit more about proteins and one important property of proteins. The property is the protein flexibility. So I talked about the way of representing protein by their sequence or the structure, but it's not the full picture, because proteins can exhibit dynamics. They can move. They can change their shape, and this is actually a very important property for the protein function. By the function, we often mean actually the ability to bind or unbind different molecules. Here we should see an example. Okay, it doesn't work, but we have the red protein, which has a flexible part, the green loop, and this green loop, it can move, which can lead to the occlusion of the binding site, which leads to the release of the yellow ligand, so the yellow molecule which is bound to it. And this motivates us to study protein flexibility, and I will tell you more about 2 challenges that we try to address in this regard. That is, one of the challenges actually to define the flexibility properly so we can study it and develop tools for studying it. And the second challenge is actually to develop a predictor of protein flexibility. So we would be able to predict which parts of the protein move in which way. And that's what we did in our work. So we developed a tool called FlexPert. FlexPert is a neural network-based predictor, and it takes on the input the protein sequence or the protein structure, and it outputs the prediction of the flexibility of the individual amino acids within the protein. To train FlexPert, we use data coming from the molecular dynamics simulations, in particular the Atlas MD dataset, which contains simulations of almost 1,400 proteins. And these molecular dynamics simulations are actually a great way to estimate the protein flexibility. The problem with it is that it's super slow. So it can take weeks to simulate the protein dynamics using molecular dynamics. And that motivates us to develop a neural network-based predictor which would be much faster. So we developed two versions of the predictor. The first one is called FlexPertSeq, and that's working with the protein sequence. So it takes on the input the sequence of the protein and outputs the flexibility for each of the amino acids. And to develop this predictor, we work with protein language models. That is the language model that I guess people are familiar with. But these protein language models, they are pre-trained on the sequences of proteins. So they know the grammar, let's say, of the proteins really well, and they are able to represent the proteins efficiently. So we start with a pre-trained protein language model, and we fine-tune it using our data on flexibility in order to get this predictor. Then we have another version of this predictor which takes on the input not only the sequence but also the protein structure. In order to achieve this, we take the FlexPertSeq predictor and we combine it with anisotropic network models. These anisotropic network models are a simplified mathematical model of the protein dynamics, which are also very fast. And we combine them in this combined FlexPert3D predictor, which takes on the input the sequence and the structure, and then again outputs flexibility, which considers both of these modalities from the input. These are the results, so like the numbers I'm presenting are the Pearson correlation of our predictions to the ground truth, which is the flexibility from the MD simulations. And we can see that both the FlexPertSeq and the FlexPert3D outperform its counterparts in terms of this metric. So there's some visualization of the flexibility predictions, but I will talk about that a bit more later, so let me move on. We published our work in ICLR, which is a prestigious machine learning conference, and we presented it this year at the conference in Singapore. The code, data, and models are freely available. And in order to make this tool even more accessible to the community, our student, Jan Richter, is currently working on a web server for this tool. The web server is almost ready, so we can have a look at a demo. So in the web server, can we have a look at the demo? No? Ah, is it? Can you play it? Oh, okay. Never mind. So, yeah, I mean, it will be anyway very soon available online. So, now to conclude, let me briefly talk about our ongoing work on applying the tool to the APOE protein. Here we can see the visualization of the prediction for the 4-helix bundle of the APOE protein. So that's the best studied part of this protein. And in the warmer colors, we can see the higher flexibility, and like in the blue-like colors, we can see the more rigid parts of the protein. But what is more interesting for us is to compare the differences between the different variants of this protein. In particular, that's what we present here. So in this plot, on the y-axis, we can see the predicted flexibility, and on the x-axis, we can see the residues of the, like the indices of the residues of the 4-helix bundle of the protein, of the APOE protein. So the 4-helix bundle is there, the structure is shown there. Now the color is not coding flexibility, it's just a legend for the different parts of the protein. So we can see the dark blue helix is, for example, would be this region in the plot. And then we see the different variants in the different contours. So the orange one would be ApoE4, and the blue one would be ApoE2, and the red one would be ApoE3. There is some shift in the absolute values of the flexibility, but we don't consider that to be as relevant. What we are interested here is actually to see relative shifts in the flexibility. And we can see the profiles of the flexibility for these three variants are super similar, which is not surprising because there's just tiny differences between these proteins. And, but where we can actually see a relative difference is at the locations of the mutations. So for the ApoE4 mutation, we can see that for the orange curve, we can see that the flexibility is relatively increased when compared to the red and blue curves representing ApoE3 or 2 respectively. Similarly, at the location of ApoE2 mutation, we can see even bigger effect in terms of flexibility, that the mutation of ApoE2 is decreasing the flexibility as opposed to the case of ApoE3 and ApoE4. So this is, these are the results we are currently working with. We want to interpret them further to find out whether this is really a significant difference. And for future work, we would also like to analyze the flexibility of the full-length ApoE protein, and not just this 4-helix bundle, because we believe we might find something relevant there as well. So thank you for your attention, and thank you to my collaborators. Yeah.
Thank you very much. Maybe if there is some space for questions from the audience. And I don't see any. Yeah?
Thank you for your presentation. And I want to ask about how long did you work during this project and your research? How long was your team?
So I would say the paper for the flexibility predictor is like, I don't know, overall, let's say 1 year, including the publication, maybe like 6 months of intensive research, I would say.
Okay, thank you. And next, and what kind of issues did you have where you work with at the level to the pre-processing with data, preparing this data? Maybe do you remember? And always we can find something.
Yes, so actually the final decision to use this Atlas dataset helped us a lot because it's a very nice dataset which was nicely pre-processed. So we could sort of dodge these notorious issues. So in the end it was okay, the data, but this dataset was released just a few months before we started this project. If it was not for that and we would have to compile our own data set, there would be much more hassle. Yeah.
Thank you, because so interesting this topic. Thank you.
Thank you.
Thank you for question. And anybody else would like to ask? Yes?
So I was wondering, from the computational viewpoint, where is your bottleneck? Is in the storage, in the access to the data, in computing power? Where do you see need of more infrastructure?
I would say it's definitely the data. So we are working here with a data set of 1,400 simulated proteins, and for each protein I can imagine the simulation to take even weeks. So, of course, you can parallelize for different proteins, but still you need a lot of compute for that. Also but it's not just compute. For the molecular dynamics, you also have to have some expertise. So you still have to have people who will set up sort of the simulation for your system, run it, and compile it in a data set. So that's why we have a data set of like 1,400 proteins, not like hundreds of thousands of proteins, I would say. So the data is the biggest bottleneck, and the reason for that is like combination of compute intensity with the need for experts. Yeah.
Thank you. Any other question? Okay, thank you very much. And I'm sorry to the people behind the cameras that we moved from podium to a completely different place. Thank you very much. Yeah. And now I would like to welcome the second speaker, which is Dr. Ninon Burgos from Paris Brain Institute and CNRS. And her topic is AI for brain image analysis in neurodegenerative diseases. So the floor is yours.
Thank you, Lenka, for the nice introduction. So yes, I'm Ninon Burgos. I'm a CNRS research director. So CNRS is a French National Center for Scientific Research, and so I'm based at the Paris Brain Institute. And so I will present our work on AI for brain image analysis in neurodegenerative diseases. So neuroimaging is a very rich data where you can have information about the structure and physiology of the brain. But sometimes neuroimaging data are difficult to analyze because they include a lot of information. And so we can use computational methods relying on machine learning to analyze these images. For example, we can use this type of approach to predict the diagnosis. Here is an example of a positron emission tomography scan, so PET scan, that shows you the brain metabolism, so how much sugar the brain is using. We can feed this type of image to a convolutional neural network that will output a diagnosis, Alzheimer, no Alzheimer. There are many approaches that have been designed to do this type of things, but very little are translated to clinical practice. This is because of many reasons that I won't have time to detail here. Instead, as an alternative, we propose to develop methods on anomaly detection. So what is the principle? The idea is to detect anomalies from an input image by reconstructing what would be the healthy version of that input image. And this can be done using a generative model. So the principle, you first have a training phase, during which you use images of healthy subjects to train a generative model to reconstruct images of healthy appearance. Then, during the application phase, you feed an image that may or may not include anomalies to this trained network that will reconstruct an image that is pseudo-healthy, because it is the only thing it knows how to do. Then what you can do is compare this reconstructed image with the input, and this will highlight the anomalies. So in the team, we work a lot with variational autoencoders, which are a type of generative models. A difficulty that we have in our context, so the study of dementia, is that we don't have, in most of the cases, a mask of the anomalies. We don't have a ground truth. So one of our first contribution has been an evaluation framework that relies on simulating an abnormal image. Because basically, without this, we can only check whether when we feed an image of a healthy control, that the reconstruction also appears healthy. This is the only thing we can do. But with the simulation framework, we can actually validate a large number of generative models because we can ensure that we can assess the quality of the reconstruction, but also assess to which extent what we reconstruct is healthy. And also we can compute anomaly scores, both on simulated and real images. So we use this framework to compare a really large number of variants of the variational autoencoders. And until then, this had only been done on in computer vision in general, but not for medical images that are usually like 3D. And here we aim to detect anomalies that are quite subtle and diffuse. We are also interested in how to handle variability that arises in pseudo-healthy reconstruction because of the variability that exists within healthy controls, but also a variability that arises from the modeling itself. So first, we proposed a way to explicitly model variability that exists within healthy controls. So we explicitly modeled this within our reconstruction process. And we also investigated the variability that comes from the fact that that arises when you use different random seeds when you train models. And actually here you can see that the performance in terms of anomaly detection varies a lot depending just on the random seeds, which you don't want. And so we show that by combining different models, we can actually have a more robust performance. My screen is off. If it can be turned back on, please. Okay. So until then, I showed you work with quite standard generative models. But we also in the team propose new, more advanced generative models. This is the case with Bayesian flow networks. So you may be more familiar with diffusion models, which principle is to progressively denoise an image. So Bayesian flow networks, they also rely a bit on this principle, but they operate on the distribution theta t of the, so on the parameters of the data distribution, rather than the data itself, x. And it uses Bayesian inference, it uses Bayesian inference to update these parameters. And it does this in by relying on the previous parameters, but also on the noisy data x. The problem with these Bayesian flow networks, the initial approach, let's say, is that it's not meant for conditional generation and really not for anomaly detection. So we proposed different contributions to make this approach work for anomaly detection. So we start from an input image instead of starting from noise. And also we change the type of noise. So we use simplex noise instead of Gaussian noise. We also change the accuracy schedule to be in a high noise regime, and also we change like the Bayesian update to always go back to the input image, again in a recursive manner. Okay. And here are some examples of results that rely on the evaluation framework that we proposed. So you have a ground truth, so the image of a healthy subject, that we use to generate an abnormal scan, and we know the anomaly that we generated. Here is the results that you can get of pseudo-healthy reconstruction with a standard like Bouteiller VAE and the anomaly map associated. So the reconstruction is not really subject-specific. It's quite smooth and it looks like any brain. Here are two other approaches based on GAN or a diffusion model. And here is the results that we get with our anomaly Bayesian flow networks. So the reconstruction looks more subject-specific, and we're able to detect the anomalies that we had generated. Okay, so until now, what I presented was developed using images acquired as part of like research. But what happens when we want to predict a diagnosis with images looking like that? Or if you want to detect anomalies from images looking like that? These are not so nice images, but this is what is being acquired every day in hospitals. So these images, they come from the one of the 39 hospitals of the Parisian area. So this network of hospitals is called APHP. And they're all gathered in a data warehouse. So this data warehouse was created in 2017 and is actually the first in France that is allowed to share data for research purposes. It includes administrative data, medical reports, imaging exams, biological results of more than 10 million patients. And so a potential of 10 to 100,000 of brain images. So as I was saying, in our community, many approaches are developed and tested with research data. So here our objective was to see how these approaches perform with real-world clinical data. Here again, we studied both diagnosis prediction and anomaly detection. For diagnosis prediction, we work with T1-weighted brain MRI, and we wanted to detect dementia patients from patients that are either no dementia with or without other lesions present in the brain. The results, not really surprising, the performance is much lower in this context than in a research context. But some observation, we show that the difference in proportions of images with a contrast agent, gadolinium, or with different quality levels was really biasing the results. We had a phenomenon of shortcut learning. And also we saw that the diagnostic codes, so ICD-10 codes, that we're using as labels were really not robust. So instead, we moved to an approach that doesn't require this diagnostic code as labels, and we go back to anomaly detection. Here we wanted to detect white matter lesions from FLAIR images. Here an example of results. So the lesions are in purple, and we had no way to have a pixel level annotation. So instead we use a Fazekas score as proxy for the evaluation that basically reflects the amount of lesions we have in the brain. And so we can see that the Fazekas score is consistent with the volume of the lesions that we automatically detect. So actually we did a lot of work with this data, a few conclusions. So really quality control and dataset homogenization are really crucial to enable the translation of computer-aided diagnosis tools to clinical practice. Also, not all AI approaches are appropriate for use in clinical practice. And also, the last point is not because you have a huge data set that it is gold, basically. Other characteristics matters. Okay. Before I finish, I want to briefly mention 2 open source Python libraries that we developed for neuroimaging processing and analysis with deep learning. So Clinica for image preprocessing and data management in general, and Clinica DL for deep learning experience. And with that, I want to conclude on the fact that by developing methods that are able to analyze images at the subject level, by developing open source software that are in favor of reproducibility, and by evaluating on real-world clinical data, we are making progress to translate computer-aided diagnosis tools to clinical practice. I want to thank the people I work with and you for your attention.
Thank you very much, Ninon. It was very interesting. I wonder, there are no questions on the Slido, but I'm sorry, we have space only for one question now. So it looks like Adam is lucky, one. And of course, if you will have any other questions, we will be very pleased to connect you to Ninon directly, so you can ask after the meeting.
It's very nice work, thank you. And I liked the part where you really looked at the robustness of your methods and if you like, the deterministic nature or not, depending on things like initial random seeding and other details. We want our results to be sort of reproducible in the sense you put the same data in, you get the same result out. My comment or question is about the final output that you were kind of looking for, which was a binary, almost like a diagnosis. And I'm wondering if that's maybe a bit simplistic and that's really something that the clinician at the end of the day is responsible for, but you could have some sort of data reduction of a complex image or a set of data that gives information that is helpful to the clinician. It's more maybe probabilistic in some sense. And I'm thinking about Václav, if he's here, and his notion of data embeddings, somehow reducing high-dimensional information into sort of simplified, but not too simple, but things that are meaningful. I just wondered if you guys have been thinking along those lines.
Yeah, well, I couldn't agree more. Actually, if we perform this diagnosis prediction task, it wasn't really for the clinical application because we know that it's basically useless. It was more we were addressing our community of so medical image computing because this is a task that is that people love, it seems, and our message was basically to tell our community to stop saying that our algorithm is ready for clinical practice because it isn't. And so it was really to mimic like the task that is done in research and just to say it doesn't work and it's useless. So it was more for like these reasons, and now we move more like towards what is hopefully like useful working with clinicians that tell us you should do that or that. But yes, I agree. This task is useless.
Yeah, it's super important because as you showed, the real world is very messy and it's completely different than a very sort of highly rarefied, maybe single data set research development. But also, of course, the issue of interpreting an image, there's just one data point in isolation. And of course, you're missing the clinical context that goes along with that. And it reminded me back some years ago when I was involved in the first phase 1 studies of donanemab, which is now approved in the US as an anti-amyloid treatment. And this was a phase 1 study, and so we were doing lots of imaging, and we were looking at these images as they came in, and we had amyloid, and we had tau, and we had structural MRI. And so we were sort of looking at each of these, and my God, what a box of frogs. There was, and this was a well-defined, clinical trial population, but there was some had a head full of tau, massively positive tau scan, and their MRI looked like a 20-year-old. This was not a sign of atrophy, and vice versa. People's MRI looked like Swiss cheese, and they were tau negative. Okay, they were all amyloid positive, so it's very variable, and so you need the broader there's more stuff going on there than just the image.
Yeah, well, it's part of our future plans.
Yeah. Excellent.
Thank you very much for question, and thank you very much, Ninon, again for your time and a lovely presentation. Now there is another last one, but very important one, a person who is coming to present CLARA research project. And while he was creating the name of the project, he got inspired by Prague, obviously, because the name is Data Alchemy, transforming multidisciplinary brain research data into actionable knowledge using computational modeling and cutting-edge technologies. And it will be presented by Jean-Marie Bouteiller.
Thank you. All right. Thank you. So I know I'm in a dangerous position because I'm the only thing separating you from your lunch. I will try and keep it short. I also know that the title is a mouthful, but my intention here was to sensitize you on a lot of aspects of the complex problem we're facing. And one of these aspects is the fact that there's a lot, well, we've been studying the brain for many years. And there's a lot of information that came out of these efforts. Now, we've been studying for a long time, but it's still unclear how it works. I think there have been a lot of presentations that indicated that we're not quite there yet. For the patients, there is a very limited number of solutions, therapeutics, that are available. Now, why is that? Well, because the brain works at many scales. We've heard about molecular interactions. We've heard about things happening at the individual level in the entire brain. There are things that are only palpable at the population level. So we have all of these things happening at the spatial scale. There are also different issues taking place at different time scales, right? Actually, I could argue that your brain now is different than it was 5 minutes ago because you've been exposed to something new, right? So if someone took your scan with the synaptic granularity, they would see differences. So there are all of these time scales that are different. Similarly, you have the same brain that you're going to have for many years ahead, or a slightly changing brain. But so whenever you're being scanned, you're only going to have a snapshot, something that's going to be static. So it's a hard problem to start predicting how things are going to evolve. So in addition to that, there are issues in the different modalities. The way your vasculature works is going to affect your brain health. Your metabolic states, your metabolic health is going to affect brain health. Inflammation, genetics, all of these aspects do have an impact on brain health. So all of these disciplines should talk to each other, should be some kind of information piece that is going to be useful in understanding how the brain works. And again, so the brain is complex. Brain is a nonlinear, causal, dynamical, multivariate system. Again, a mouthful, not for the fun of saying it, it's just, it is hard. So all of these, again, are studied at multiple scales, and within CLARA, there are different research packages that each one of them is trying to address one of these issues, and then the research package 3 is aimed at trying to bridge them, bring them together to extract as much information and create models that would allow us to really get a complete dynamical understanding of that dynamical system that's the brain. So again, I'm going to hammer that complexity issue, because as you talk to different people, they're going to have a different way of looking at how healthy a brain is. You know, it could be a lifestyle, it could be physical metrics, it could be structural metrics when you look at MRI. It could be molecular markers when we talk about biomarkers. All of these aspects give you a point of view, but not really the full picture. Multiple disciplines. Well, being a neuroscientist can mean very, very different things, right? So you have all of these disciplines that each one of them is going to be focusing on one specific aspect. That's not all. You also have disciplines that intersect with neuroscience. And all of these, well, they have produced, in many decades, a lot of information, a lot of data. Now, roughly 300 to 350 journals linked to neuroscience. Over 2.4 million brain-related articles published. And again, this is not going to stop here, right? It's only increasing. So how can we go? I mean, would it be possible that there is knowledge in those papers? Is it possible that, you know, we just haven't really looked deep enough, or we haven't integrated this information well enough? Now, in addition to papers, well, you can say, well, there is data. We can use data. Well, it's true. There are a lot of databases. You have your imaging databases, genomics databases, you know, you name them. And actually, there's so many databases that nowadays you kind of need to have a database of databases, right? So, again, how can we go about getting some clarity within all of this? Well, there have been some efforts in trying to integrate all of that. So there are newer standards that have evolved. One of them is at the laboratory level is called FAIR. FAIR means findable, accessible, interoperable, and reusable. So that data sets are not just dying, but they can actually be useful for other labs and potentially integrated in something bigger. One of the aspects is that this data can then be placed in repositories. And again, repositories are a good way of organizing the data. Similarly, these repositories, they are searchable, right? So there are search engines can look at them, index them, and provide an effective way of searching the data. But it's still not easy. It's still something that Olivier was mentioning yesterday. You know, the modalities by which the data was acquired is a little bit different. And then the metadata that was used is a little bit different. So integrating these things is like, you know, a puzzle with pieces that just won't fit together. So it makes things a little more complicated. So one way to go about this would be to use newer tools. The newer tools that have been discovered, could they help us to integrate information found, for example, in publications? Well, so what we decided to do was to create some proof of concept. Would it be possible to aggregate information from the literature? Yes, it's not going to, it's not going to be clean. Every paper is going to be different. Authors are going to use different ways of presenting their data. But let's see what we can go about it. And so we used a very simple LLM, large language model. It's local. It's not even on the cloud. It runs on the laptop. And we propose to enhance its context base using what's called RAG, or Retrieval Augmented Generation. We basically force feed the duck. We're providing information from these papers and having the large language model ingest these papers. And in addition to that, we added what's called a knowledge graph. So we had the LLM generate a knowledge graph from these publications. What does it mean? Well, a knowledge graph is kind of a structural representation of how entities within the data are connected with each other. So how are they connected? For example, for people who know the movie Casino, well, it would mean that movie, in that movie there were several people that acted in it. Robert De Niro, Sharon Stone. So you now have entities, movie, actor, that have a relationship between them. So in doing that, you can start creating connections between different aspects. So why knowledge graphs? Well, knowledge graphs are actually a good way of organizing information. It helps provide context. It facilitates a deeper understanding. And it also allows for structured interconnection between information. And basically, it allows you to if you imagine that you have multiple papers that mention different aspects of the disease, well, you could now link papers that originally seemed completely unrelated, right? Because they share some notes. So it really supports what's called multi-hop reasoning, because you can generate these longer interconnections. So again, proof of concept that an undergrad in computer science generated was a very small, large language model that contains this graph RAG that was augmented with this graph RAG. Everything local, local database, local large language model, only 3 billion parameters, so starts relatively dumb. I mean, it's really unsmart. But so you can access it with a web interface, and we built a few agents to interact with it. And we only used 12 publications. We fed it with 12 publications that focused on some cellular mechanisms, some cell-type specific cell responses, neurons, astrocytes, microglia. All of these cells kind of interact with each other in the nervous system. And then we looked at, we also incorporate some temporal dynamics. All these papers are here, and they provide a different view of the cell life and activity. And then we asked questions to the system. And we asked questions and tried to emphasize some integration aspects. Was the LLM capable of creating bridges? And what we saw, so not simple questions, right? It was questions that were relatively complex. And in response to those questions, the graph focuses on one aspect. Here, so we only include 45 nodes for visualization purposes, but 12 papers resulted in 16,000 nodes, 39,000 relationships, right? With 12 papers. So we can generate all these connections, these interconnections. And then we evaluated that the response of the system with different evaluation metrics using another LLM or another Python frameworks for the evaluation of large language model responses. And we compared that to expert verified responses using 3 criteria: correctness, coherence, and relevancy. And we computed the average. Now, here, again, very preliminary result. Very small LLM, very small number of publications. But, so the naive model, we did not even graph because it was impossible for this model to answer the question. Now, the RAG, if we did not include the interconnection and the knowledge graph, we only used the augmented system, provided roughly 60% of score, overall score. Well, the RAG graph on these 12 layers was already capable of increasing about 10%. Again, very simple system, right? So we believe that using these newer technologies like really allow to get a more complete understanding. There are so many aspects within neuroscience, within how we look at the nervous system, that could these tools be useful to help us get there, get a more complete understanding? So, long story short, I have no time left. So, again, there's a lot of information that's out there. A lot of scientific evidence is available. So, without digging further in the ground, could it be that the pile of dirt that we have extracted already contains useful knowledge that we have not yet extracted? It is possible. We have novel methodologies, new tools that could help us integrate that information and extract knowledge. This could be done across sources, scales, and disciplines. I think it'd be useful to try and do that because it would really yield a more comprehensive understanding of what is happening in the brain. So we just developed, again, a very small proof of concept, and we intend on moving further with that. And again, this is an open problem. Publications are available. We can do that as a collaborative work. Preliminary results are encouraging, and the proof of concept can be considerably improved. Considerably improved. There are a lot of techniques that can be applied to that to extract meaningful knowledge. So it can facilitate knowledge extraction. It can generate a much more comprehensive map of what is happening in the brain. And in the end, it should allow us to really focus on where can we intervene, where can we get, where can we make a difference, either to maintain the health of the brain or to have a therapeutic intervention that will make a difference into improving the symptoms. Yeah, and thank you very much for your time.
Thank you very much, Dr. Jean-Marie Bouteiller, for this great talk, representative of CIIRC for this part. We have got, again, only one question, space for one question. Yes, thank you. Ira?
I was wondering if you are closely working together with the cancer field, because there they have a tradition for this searching information, as cancer is as big as neuroscience.
Right.
And there are great developments during the last years. So I was wondering how you are interconnected.
I've been following that, and I think they have a tremendous amount of knowledge in these kind of knowledge integration field. So I think, again, both the NDRC, CLARA, and the scientific community should work openly, because that's how problems are solved. I think that there's one thing that I really like is none of us is smarter than all of us, and that's the kind of meaningful answer that we're looking for, that can definitely benefit from very open collaboration.
Thank you very much. Thank you very much, Jean-Marie, again. And now I would like to invite you all for a lunch and some break. And if we could start again at 1:10, it would be lovely, because we had some kind of like a smaller gap for lunch that we were expecting to have. But please, we have some more interesting topics in front of us. Thank you very much, and enjoy your lunch.